Now we are going to learn about messenger RNA or mRNA. mRNA is also known as messenger RNA, is a type of RNA molecule that carries the code from the DNA in the nucleus to the site of protein synthesis in the cytoplasm, that is ribosomes. mRNA is the version of the genetic material that leave the nucleus and move to the cytoplasm where responsible proteins are synthesized. So the information in DNA cannot be translated directly to proteins. It must be transcribed into its RNA form called the mRNA that now encodes for specific proteins. This process of transcribing the information in DNA to mRNA is called transcription, which is catalyzed by the enzyme RNA polymerase. mRNA, being an RNA, is a single-stranded molecule, and the length of ribonucleotides present in mRNA can vary according to the regions of the DNA transcribed, considering the type of proteins needed by the cell. With each 3 nucleotide base sequence in the mRNA molecule, one specific amino acid is coded and subsequently a chain of amino acids or proteins is obtained by decoding these basic sequences in the mRNA molecule. These codes are in the form of triplet sequences of nitrogenous bases and are often referred to as codons. mRNA definition, properties, structure, processing types, functions, etc. are given after this. So, messenger RNA or mRNA properties. mRNA accounts for about 5% of the total RNA present in the cell. The length of ribonucleotides in an mRNA molecule varies widely depending on the type of protein they account to code for. Therefore, the mRNA molecule is considered the most heterogeneous of the 3 types of RNA present in the cell. mRNA is synthesized from a DNA molecule by reaction of enzyme RNA polymerase, and this process is termed as transcription. At the end of transcription, mRNA is synthesized. This newly synthesized mRNA is further edited or modified to remove non-coding regions and also to make the molecule stable. This process is called post-translational modification and takes place only in the eukaryotic cell. During post-transcriptional modifications, along with the cleaving out of non-coding regions from mRNA molecule, they are capped on their 5' end with guanosine triphosphate nucleotide to make it recognizable to the ribosome during protein synthesis. Additionally, during the post-transcriptional modification process, the 3' end of the mRNA molecule is also edited by adding numerous adenine monophosphates at their ends, also referred to as poly-A tail, which makes the mRNA molecule more stable by preventing enzymatic degradation. mRNAs are sensitive to heat and can be degraded on prolonged exposure to high temperature. However, for some manipulations, they can be heated for a specific period of time at distinct high temperatures. So here we could see a picture of prokaryotic mRNA. The 5' end is there, 5' UTR regions and translated region. Shine Delgarno sequence along with start and stop codons, protein 1. Just remember this term of Shine Delgarno sequence, don't forget that. Okay, protein 1, protein 2, start stop regions of protein 3 along with 3' UTR and translated region. Later you could see a picture of eukaryotic mRNA which shows 5' M7 G loop GPPP cap, then UORF region, 5' UTR region, IRES, IRE, start, AUG methionine, UAG, UAA, UGA. Any of this code for a stop coding sequence or a ORF region. Along with that, IRE and miRNA, 3' UTR binding elements, 3' UTR polyadenylate tail. Okay. Next, structure of mRNA. mRNA is a single-stranded RNA molecule running from 5' to 3' direction. In a prokaryotic cell, there is a lack of distinct nucleus. Thus, the mRNA synthesized contained a copy of DNA sequence within the terminal 5' triphosphate group and 3' hydroxyl group. Prokaryotic mRNA at its 5' end has a Shine Delgado sequence which is rich in purine nucleotide and is essential for facilitating the binding of mRNA to ribosome. Prokaryotic mRNA starts at 5' with a triphosphate group followed by Shine Delgado sequence or untranslated region, and as we move along to the 3' direction, the coding regions begin. The coding regions begin with a start codon and end when it reaches a stop codon. After encountering a stop codon, protein coding regions come to an end, after which there is another untranslated region that terminates at the end of 3'. However, in the eukaryotic cell, the nucleus is distinct and has numerous enzymes in it that make the synthesized mRNA molecule go through modifications at their terminals 5' and 3' to maintain the integrity and stability of the mRNA molecule. There is post-transcriptional modification. The 5' triphosphate group of eukaryotic mRNA is esterified, forming a cap structure. This process is referred to as 5' capping, which happens when a 5-methyl guanosine cap is added to 5' free triphosphate group via 5' phosphate linkage with the help of the enzyme called guanyl transferase. The 5' capping is important for the recognition of mRNA by the ribosomes during protein synthesis. And likewise, the 3' hydroxyl group is cleaved to give a free hydroxyl group to which numerous adenine monophosphates are added to make the mRNA molecule more stable and prevent it from degradation. This tail of numerous adenine nucleotides is referred to as poly A tail. The poly A tail added to the 3' end of the eukaryotic mRNA are usually 100 to 200 bases long. The addition is catalyzed by an enzyme poly A polymerase that recognizes the sequence AAU-AAA as a single for addition. The poly A tail is vital during transporting mRNA from the nucleus to the cytoplasm for protein synthesis. Eukaryotic mRNA acts at 5' with a cap followed by untranslated regions 5' UTR, and as we move along 3' direction, the coding region begins with the start codon and continues till it reaches the stop codon, which marks the termination of coding region, after which there is another untranslated region 3' UTR that terminates when poly A tail begins, which further terminates at the end of 3'. Okay, so here we could see eukaryotic mRNA structure. The structure of a typical human protein coding mRNA, including the untranslated regions or UTRs, cap region 5' UTR, start coding sequence or CDS, stop 3' UTR, poly A tail regions. So these are the major regions. Then messenger RNA composition. mRNA is a biopolymer of nucleotides bonded with each other via a phosphodiester bond. The number of nucleotides in mRNA varies widely according to the type of protein it can encode. The nucleotide that makes up the mRNA are also referred to as ribose nucleotide due to the presence of ribose sugar in their structure. Overall, mRNA is composed of ribose sugar, phosphate, and nitrogenous base. Ribose sugar is a cylindrical structure made up of 5 carbons and 1 oxygen atom. This sugar contains 2 OH groups at 2' carbon and 3' carbon. This ribose sugar is attached to a nitrogenous base via hydrogen bonding. There are 4 nitrogenous bases, namely adenine, guanine, uracil, and cytosine. These nitrogenous bases pair complementarily with each other, G with C and A with U. A mature mRNA in the eukaryotic cell also consists of a modified 5' end with a 7-methyl guanosine cap on it and a mature mRNA in the eukaryotic cell also consists of a modified 5' end with a 7-methyl guanosine cap on it and a modified 3' end with polyadenine nucleotides on it. Eukaryotic mRNA before post-transcriptional modification is called precursor mRNA, having both non-coding regions or introns and coding regions of exons, and no 5' cap and 3' poly A tail on it. But after post-transcriptional modifications of precursor mRNA of the eukaryotic cell, the non-coding regions are cleaved out and 5' capping and 3' poly A tail are present on mRNA, thus making it a mature mRNA ready for protein synthesis. However, in a prokaryotic cell, no such post-translational modification takes place, so the mRNA synthesized has both coding and non-coding regions on them. Additionally, no capping and no poly A tail are observed, making them prone to degradation. We now can say that the composition of mRNA depends on the type of cell they are in and in what phase of their processing they are in. So messenger RNA processing. mRNA processing refers to the steps that are carried out before mRNA is finally ready for protein synthesis. When mRNA is synthesized from the DNA molecule, the mRNA we will get is a precursor mRNA having the same 5' triphosphate and 3' hydroxyl terminal residues as that of the DNA. This newly transcribed pre-mRNA is prone to degradation. So to predict it, this pre-mRNA is coated with RNA stabilizing proteins as soon as they are synthesized. Not only that, but the stabilizing proteins are also responsible for the safe delivery of mRNA from the nucleus to the cytoplasm. This precursor mRNA processing happens in 3 important steps. First step, addition of stabilizing and signaling factors at 5' and 5' capping. When pre-mRNA is being synthesized, its 5' end is immediately capped by a 7-methylguanosine cap via 5' phosphate linkage. This cap structure protects the mRNA from degradation, and in addition, they also help in the recognition of the mRNA molecule by initiation factors involved in protein synthesis to initiate translation of ribosome. Next, addition of stabilizing and signaling factors at 3' end, poly-A tail. The newly synthesized mRNA from double-stranded DNA with the activity of an enzyme RNA polymerase is premature to initiate protein translation, so to make it mature. In addition to 5' capping, 3' end of a pre-mRNA also needs some modifications, and this is carried out by an enzyme that cleaves the 3' end and adds numerous adenine nucleotides to it, thus making a poly-A tail at the 3' end of the pre-mRNA. The number of adenine nucleotides added is said to be about 200 approx. This poly-A tail prevents mRNA from degrading and plays a vital role in transporting mature mRNA from the nucleus into the cytoplasm for protein synthesis. Then precursor mRNA splicing. Genetic information transcribed from DNA molecules contains both introns and exons. But for mRNA to be translation ready, it must only have exons. So we have to splice these introns out and keep only exons. This process of removing introns and keeping only exons in the mRNA sequence is called mRNA splicing. The process of splicing occurs by recognizing a specific sequence that initiates the cleaving out of the introns and rejoining of exons with utmost precision and accuracy. The splicing process is made possible by complexes of proteins and RNA molecules called spliceosomes. Each of the spliceosomes consists of 5 subunits called snRNPs for small nuclear ribonucleoparticles. These snRNPs are only found in the nucleus. snRNP, small nuclear ribonucleoparticles. These spliceosomes recognize sequences at the 5' end and 3' end of the intron. The specific sequence they recognize are GU nucleotide at the start of 5' end and AG nucleotide at the end of 3' end. Now that the spliceosomes recognize the introns, they cleave them out by breaking the sugar phosphate of pre-mRNA at the 5' end of introns and covalently attaching it to a nucleotide internally within the intron. This results in cleaving out of introns. Spliceosomes now connect the free 3' end of one exon to the adjoining exon with a free 5' end, thus making the joining possible. Now the mRNA that we have is a mature mRNA with 5' cap, 3' poly-A tail and only exons. So capping is here, mRNA capping enzyme, guanylyl transferase, guanylyl T-methyl transferase, etc. Then A capping, then B splicing, splice site exon, U1 intron, branch point, polypyrimidine tract, 3' splice site, transesterification, guanine 7-methyl transferase, then cleavage and polyadenylation. Cleavage is created, then cleavage is happening. After that, polyadenylation, poly-A part will be added. Okay. In which region? 3' region. Then A to L editing, A to I editing, whatever it is. Ribose, adenosine, etc. can be seen. Inosine. Next, types of mRNA. Precursor mRNA. It is a primary transcript for a mature mRNA as it comes off the DNA template. The pre-mRNA contains sequences that are removed before translation. These sequences are removed by the action of complexes of protein and DNA molecules called spliceosomes. In addition to the removal of sequences, some modifications are carried out at 5' and 3' ends respectively. After all, the unwanted sequences are removed and the 5' and 3' ends are modified. It is ready for translation. Now we term it as a mature mRNA. Then monocystonic mRNA. This type of mRNA molecule contains exons regions that code for a single protein. All eukaryotic mRNA is monocystonic and they all go through post-translational modifications to initiate protein synthesis. Then It is transcribed from a single gene or cistron and has one initiation and termination codon. Initiation and termination codon, monocystonic. And polycystonic mRNA, in contrast to monocystonic mRNA, this molecule of mRNA encodes for more than one protein. Present in prokaryotic cell and also in the chloroplast of plants, it is transcribed from more than one gene, and this has many limitations and termination codons. So we learned about precursor mRNA. This is the primary transcript for a mature mRNA as it comes off the DNA template. The pre-mRNA contains sequences that are removed before translation. These sequences are removed by the action of complexes of protein and RNA molecules called spliceosomes. In addition to the removal of sequences, some modifications are carried out at 5' and 3' ends respectively. After all, the unwanted sequences are removed and 5' and 3' ends are modified. It is ready for translation. Now we term it as a mature mRNA. Later is about monocystonic mRNA. This type of mRNA molecule contains exon regions. Exon in the sense they are coding portions that code for a single protein. All eukaryotic mRNA is monocystonic and they all go through post-translational modifications to initiate protein synthesis. It is transcribed from a single gene or cistron and has one initiation and termination codon. Then polycystonic mRNA, in contrast to monocystonic mRNA, this molecule of mRNA encodes for more than one protein, present in a prokaryotic cell and also in the chloroplast of plants. It is transcribed from more than one gene and this has many initiations and terminations codons.